So we just took up the homework previously. We took up the homework
on 1070.
This is the first content slide. I'm sorry, I forgot to press record. So we're just going to start recording from now, but that's okay because we have the slides and everything else. So, Dr. Smith, we're going to be talking about evolution for the next few weeks.
Hooray!
Hooray! You can go to this little website here if you would like. We can probably...
Just click on it.
Can I?
Yeah.
No, it's just on my slides. So maybe we can pop it in the chat at some point. It's super cool and super freaky to like think about how many species there are on the planet, how many species there were on the planet,
Many more,
and how they got there is to me mind-boggling. And what we want you to do is to not memorize a definition of evolution. We want you to be able to develop a bit of a word cloud when we think about evolution. So, Smith, without coming up with a definition, what words are important to you when you think about evolution?
What words are important to me when I think about evolution? Inheritance.
Okay. Okay, that's a good word. What else?
So like you get money. No.
No. Okay, what other words? Pop words in the chat too if you'd like.
Darwin, thinning of the herd.
Yeah, thinning of the herd.
Whoa.
Oh my goodness. That's a strong one.
Mutation.
Mutation. That's a good
you're
word.
going to get barraged. Adaptation, development, genetics, survival of the fittest,
Yep.
Homo sapiens,
Yeah.
genes, growth, species, speciation, descent with modification.
Yeah. Good.
That book called The Selfish Gene.
Okay. Yeah. Super good. And thank you for that contribution. That's really, really important.
Well done.
Well done. And I picked out a few of them. And what I would like to suggest is that when we ask people what comes to mind when they think about evolution, a lot of the time what comes to mind is related to a specific type of evolution, okay, called natural selection. So everything related to Darwin, everything related to survival, to adaptation, to thinning of the herd, all of those things have to do with natural selection. And evolution itself is a much bigger term. So we're pretty sure everyone kind of sort of gets natural selection. Like, we really focus on that when we learn about it.
And we know that you've learned a lot about pepper moths.
Yes.
In your curriculum, probably wherever you're from.
We're not going to talk about pepper moths.
I mean, they're amazing. Come back in 2700 next year.
Yeah, because
we'll
they're
talk about
changing
pepper moths.
back.
Yeah.
It's
amazing.
It's amazing. Okay, but we want to introduce you to like every other part of evolution that to me is more mind-boggling, more amazing. So when I think about evolution, just evolution, not natural selection, but the big term evolution, to me, the one word that captures it all is the word change. Okay? So it's right there in green in the middle of your screen. Evolution is about change. It is not about the direction of change. It is not about something getting better. That's an adaptation. It is not about something getting worse, right? That's a non-adaptive mutation. It's just about change. And if there is change at the level of the genes that is inherited, then you have evolution. It doesn't even have to be selected for. It just needs to change over time, okay? So in places where we don't see change are things like clones, right? If there's no mutation, if you have an exact clone of something from one generation to another, there is no evolution. It's very unlikely that that's going to happen because there's so many things happening where mutations can happen. And if there's a mutation, then there is change, and that does lead to evolution. Okay, so we kind of want you to kind of blow up your minds a little bit and go, oh my goodness, it's not just about, Darwin and pepper moths. It's a lot cooler than that.
Easy. There's many, many, many things as cool.
Yes.
Darwin and pepper moths is pretty cool.
It's cool. It's true. It's cool. I don't want to degrade Darwin.
And Doug loves that bone. You are correct.
Okay.
There's actually quite a few people who didn't.
Okay.
Said that they don't know what pepper moths are.
Oh, brilliant.
So I said, stick around. We can
Yeah,
talk about
we can
them
talk about
afterwards
pepper
if you'd
moths
like.
if you want, for sure. Okay. Why study evolution?
Why not? All sorts of reasons. Sometimes, particularly maybe at this point in your career and maybe at this point in what we've all been experiencing over the last 18 months, there's many reasons that pop in your brain, and they might even be ones that affect humans. They might be ones that affect medicinal care, or disease transmission, or in fact, like this David Quammen book about the transmission and the jumping of diseases from one species to another.
Yep.
That's a great book if you haven't read it.
Oh, it's good. Yeah. I feel like it's a bit too soon.
Oh, no.
But no? Okay.
No. I mean, there's gonna be a new edition, I'm sure. If you haven't seen it yet, it came out, I don't know, maybe 10 or more years ago. So the COVID story is not in it, but right. I'm sure he's writing. Yep.
Okay. There are other reasons. Check this out. I'll just press play in a second. But what you're going to watch, and they should explain it, but in case the closed captioning doesn't work well or whatever, what you're going to see is a giant agar plate, like a petri dish, but it's massive. And they've inoculated the sides with bacteria. And then they've divided the plate, and you'll see, they'll explain, into areas that have increasing concentration of antibiotics. Okay, so these are just regular bacteria. They are not resistant in any way, and you'll see what happens over time.
Picture just like a foosball table. This is a foosball table where each of them start at their own end, and as you go closer to midfield, there's an increasingly harsh environment.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pour some thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads, until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
Extremely high, and like orders of magnitude change. So not just going like a little bit 5 to 7, which I would find a problem, like going 5 to 50 and 50 to 500, and it's like that's those kinds of changes. I guess I'm most frequently an ecologist, so when I count, I don't really worry about the fact that if I didn't end up if I'm counting things running across a plate or an experiment, and if I get 6 and there were actually 7, that's not generally a big deal. If there were 60 and I counted 6, that's a problem. That's a real error. So those orders of magnitude changes, those are biologically big, crazy deals. I'm going to pop the actual paper if you want, if you're interested.
Cool. Yeah, thank you. You're up again.
So why do we think about evolution? Well, it's fine to be selfish. Sometimes you want to be selfish. Like I could be selfish and try and claim the screen. And then other times it's not good to be selfish. This is an example. We're phylogenies. We're tree-based thinking. So a phylogeny in this case is a branching diagram that where the tips, so it's displayed in a circle. You have 3 equivalent circles here that all have the same species, all have the same tips, and then those branches indicate who's most closely related to whom. And these phylogenetic hypotheses, these hypotheses of evolution, have been very useful to us to help bioprospect, as the title lets you know there. So in the colors here, basically one of the things you're looking at is from 3 different regions, from the tip of South Africa, from Nepal, and from New Zealand. Lineages in red that we use, that human societies in each of those regions have used for medicinal purposes. Now, why is that interesting? Well, we can map it on these phylogenies, and then we can find out. I was just about to do the old school. I'm going to point at the screen right where you can see.
right there.
What you can see is that in some cases, all close relatives of a red clade or a red group we've looked at, we know about. But in some cases, there is a bit of that tree that has some branches red, where we've used them for medicinal purposes, and some not. So this is an example where phylogeny can help us go, well, if 3 of the 4 relatives have biomedicinal properties, let's try and examine the 4th. And then, of course, there's what we've all been living through. And you've probably become more used to thinking about phylogenies. This is, if you haven't, this is just going to roll quickly through. When I showed this this morning, you said too soon. And I'm like, well, the very end of this graph is too soon. It's marching from left to right through a phylogeny as new lineages of COVID-19 are added and sequenced, and where they're appearing. In this case, I've simplified the figure to North America. We just passed where Delta emerged, but our phylogenetic thinking has really changed and informed how we've responded to this global pandemic that we now share and will talk about forever.
Yeah. Great. So, very, very important for all sorts of reasons, including ecological, business reasons, management, conservation as well. So, who we take out of a population of fish, for example, which fish that we're going after can have a huge impact on the actual species itself, right? So imagine if you were going after tuna, you were hunting tuna or fishing tuna, and you were only fishing tuna that were particularly large, right? And you just kept on going, fishing. Over time, those genetics, those mutations that led to these particularly large fish would be removed from the population. And what we've seen is kind of a shrinking of the individual size of fish in response to these sort of harvesting practices. And so maybe if we were going for something because it was small, then we see populations getting physically larger. So these removals of genes or of traits from the population as a result of our commercial practices has a deep effect on the evolution of the species and therefore on the ecosystem itself. But we're going to talk about it on the CourseLink site. Thinking specifically about mussels.
Now, Are you mussels?
So mussels, I honestly didn't have a lot of respect for mussels when I started teaching this course, and now they freak me out. And by the end of this class, they will freak you out.
And as we saw when you mapped for us where you are in the world, the majority of you, not all of you, but many of you are in Eastern North America and in the kind of Great Lakes lowland where we are in Guelph. And this is one of the most diverse endemic taxa groups of organisms that we have. And they are changing. And as you may guess, not for the better.
No, but the species, like the organism itself, the group itself, mind blowing.
And somebody's also noting in the chat, and they're delicious.
Also, with fries and a little bit of mayonnaise. Oh, anyway, okay. So let's think a little bit about mussels. And we're gonna, again, one of the big learning outcomes for today is to throw some vocabulary at you. Don't memorize definitions. Use your own words to create these anchors in your mind, okay? So we have a word, endemic. That's such a specific word, right?
Endemic.
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 Yeah. So what does it mean? What does it mean?
endemic.
Endemic. What does
It's
it mean?
indemnare.
Indemdare.
Indemdare, it's ick. Indem, ick. It's gross.
It means that a species in that region evolved in that region, that it is from there and therefore nowhere else. Okay, so the region can be a little tiny island. It can be the continent of North America, right? It can be anything in between. You need to be able to precisely define your region. But once you do, anything that is there, that evolved there, that is not from somewhere else and is not somewhere else, is endemic.
And so S. Sahido 3, pardon me for calling you by your first name, pandemic.
Oh.
It's the same root, but pandemic refers that it is everywhere. It's not in that spot there.
Endemic,
everywhere.
In that spot. Pan, meaning everywhere. Yeah, good. Thank you. That's great. Good connector. Okay? Yeah. So, we have a question for you. Please go to www.menti.com and I will launch the question. There you go. So the question, what factors contribute to threats to native freshwater mussel biodiversity in the Great Lakes region? Again, these are precise terms, right? So not just mussels, mussel biodiversity, which is kind of mussels, but it's like more specific. You get me a point. Okay.
Yeah.
Go. Yay.
I like a word cloud. It makes me want to fly away.
Wonderful. This is great. Yeah. Good. We'll just give you a few more seconds if you want to join in.
And you're going to swing it over there. And
Yeah.
grab it?
Yeah. Well done. Yes. All of these things. Super good. Okay. Yeah. Lots and lots of things contribute to the mussel biodiversity. Okay. So that biodiversity, we will talk more about its definition, but it has a lot to do with the different types of species or the different number of species rather than individuals within a population. And so you've got it. Invasive species affect the number of endemic species that are there. Pollution also affects generally the biodiversity, but maybe more specifically those that are more sensitive, such as freshwater mussels. So that's really, really good.
I also want to throw in again, it's an observation I made the last time we made a word cloud, but it's well done you.
Yeah.
It's a well done you observation about the diversity of words that we have here. That there are 2 or 3 that are quite big. So those they're coming up a lot of times, but you're coming up with a lot of stuff, which is we love it.
In your own words.
And I love that there's zebra mussels.
Zebra mussels. So good. Okay, that's great. Thank you. Well done. Okay, so we have a question for you. We talked a little bit about something that's relevant. Let's talk about something that might be irrelevant. So unlikely to be relevant to the conversation. And this is equally, if not more important as a biologist, to recognize what things are relevant to the conversation, to the precise language that we're using, and what things are not. So here we go. I'm launching a poll at you. Please don't annotate the slide. Please answer on the poll. And then we will take it up in a minute.
I really like the idea that differentiating the non-important thing is often what's most important, because to me that really speaks to a lot of real world experience about being in rooms where there's lots of smart people and everybody's throwing ideas around. It's like, that's kind of that, okay, and maybe a lot of these are not going to solve the problem, but you have to be able to go, that's the one.
Yeah.
Pull that one out and let's dedicate our effort to that.
Exactly. Smart.
Smart, eh?
Smart.
As they say, pronouncing the schma and the art.
Okay. You're doing very, very well. We're just going to give it another few seconds for, so 85% of you have already clicked in. That's great. We'll wait till it gets up to 90.
Somebody's asked to see Doug.
Oh.
Last time I tried, I broke the chair. So let's at least wait till later in the lecture.
Okay.
So Smith doesn't.
90% share results. Very well done. The answer is daily fluctuations in weather. Now, maybe you're wondering, well, why is that one right? Well, I got it right, but I'm not sure quite why. Try to verbalize why daily fluctuations in weather are unlikely to be relevant to explain the biodiversity in mussels. And then, if you want, and put it in the chat, and then thinking about the weather, thinking about temperature and precipitation, what word could we change it to by changing the scale of weather that would be relevant to biodiversity?
So are you saying whether or not? English is awful.
Yeah. We want to push your brains, okay? And do now, if you want to write on the slide, please do feel free.
And
so cross out the word weather.
There's some great
or
thoughts
the word
in the chat.
Yeah, the word fluctuate, whatever you want, and kind of make that a true statement that would contribute to the biodiversity in freshwater mussels.
And lots of people in the chat, you're coming up with the fact that this is a time issue. Time.
You are
Daily
amazing.
fluctuations don't have enough to impact a large scale.
Yes.
Climate. If we stick around.
Good.
Such good, yep.
Wonderful.
I love the fact that these are sentences that roll off your tongue easy, that daily weather isn't indicative of overall climate.
Awesome.
And I feel like we should be doing daily
Done.
weather isn't indicative of overall climate.
It's kind of a message that many people need to hear.
Yeah.
So you are amazing and
Thank
brilliant. Thank you. Yes.
Oh, sorry. Daily.
Oh, is it loud? Yeah, sorry. Okay. Well done. That's great.
What did we do last time that made people jump? Oh, you have to shout something.
Yeah.
Will you clap along?
Yeah, super. Well done. I think you get it. And so
Can I-
we will
can
move
I
on.
screen
Screen
grab
grab that because it is good. Yeah.
I'm having a hard time seeing it. There it is.
Have you got it?
I don't have it yet.
Okay.
I don't got it! I got it.
You got it? Okay. I'll save it too. Okay. Well done. Moving on.
We have so much time.
Yeah.
I mean, so much time.
You want to just take a break? No. We have so much to tell you right now. Okay. Now you can kind of sit back, relax, because we won't ask you to click anything. I don't think. Maybe until the end. Anyway.
We might.
Just kind of chill out and allow your brains to be blown up with awesome. Okay? It's really cool. This is my favorite part. You want to back and forth this one? Because it's awesome?
Of course, I love backing and forthing.
Okay. Ready. Good.
Those of you who asked to see Doug, though.
Oh. There's Doug.
He's asleep. Shh. Be very quiet.
And there's Lily.
Oh, yeah. I think they can see. Oh, Lily, you're licking your posterior.
Okay, good. Ready? Okay, so we're learning about freshwater mussels. There are two types of freshwater mussels. There's the unionids, and then there's the not unionids. And all of the stuff related to the not unionids you can learn about on your own time as well as on the CourseLink site. Those are the zebra mussels, okay? We want to focus on the endemic species that are in the Ontario lakes or in the freshwater lakes but because they will blow your mind. They're amazing and they are endemic, so we should learn about them. They have this very interesting life cycle, okay? They're dioecious is the word. Di, meaning two, ecious means homes. So they have two homes, okay? And one of their homes is a fish, which is just bananas to me, a fish. So they spend some of their life cycle as a parasite within the gill tissue of a fish, okay? So here we have these Glaucidia. that come out. So these are like the reproductive bodies of of a freshwater mussel that are exuded in very creative and different ways, and that's what's going to blow your mind in a minute. From the mantle tissue of the Glaucidia, they parasitize a fish host in the gills, and then they release themselves when they are juveniles, and they mature, and they settle down and become these sessile things. And now I'm wondering about, is it dioecious or monoecious?
I don't think it matters in this case. But, I mean, I think that the important thing here is just to somebody asked about the word Glaucidia. It is there. It's spelled in the chat, and it's spelled right there. Again, spelling does not count. Comprehension counts. But if you are like a word nerd, that basically means barbed. That's derived from the Greek for barbed, and that's because of the way we're going to show you a picture of them in a second. But they are an incredible stage that has evolved in these freshwater lineages native to the waters, to the rivers of Eastern North America. Yep.
So that's the cycle. They need this fish. It is critical. Without the fish, they do not survive.
So the other word that you use, sessile, is that cool with everybody?
Yeah.
So sessile just meaning not moving. In this case, not moving very much.
That's right. They move around a little bit.
Yeah.
But okay.
So this is another kind of a colored diagram of this life cycle, but put into a context and with some other examples of biology shown there. So we've got different species of mussels here down embedded on the bottom. On the stream bed, we've got a particularly grumpy looking bass or some other panfish in the back, some host species. And then we've got these blow up diagrams. So let's kind of add some words here. So we talked about Glaucidia. But take a look, zoom in, see mantle lure right there at the bottom? See that fish? That's not a fish. That is part of the mantle, which is the outer layer of the body that actually, in most cases, for mussels, it does, I mean, the mantle, come back to 2700, the mantle does, with given enough time, the mantle can build practically anything. And I'm not even exaggerating. From eyes to looking like a fish to building a shell. The mollusk mantle does it all. But that's a lure that particular species of mussel has evolved to attract a particular species of fish. Not all of the fish. So it's evolved not to look like that fish, it's evolved to look like the prey of that fish. I'm going to pause for a second so you can like pull your brain back out from your desk and put it back into your ear.
How does it know what the prey look like?
That's kind of amazing. So and there's even other strategies. So that's a particularly gorgeous one. But there's ones where they make also conglutinates. And glutinous, this is a gluing thing. So this is another product of the mantle where they imitate prey species of a fish and try and get them to come and just nibble on them. Wow is exactly right. Like wow to the 10th, 100th, 10 to the 16th. It's a mole of wows. And there, the other we see broadcasting conglutinates. That's just like a broadcasting, as you can imagine. You're going to see some video of the Glaucidia broadcasting in a second. Yeah. And then physically trapped is another adaptation of the mollusk where it's like, no, come here. I've got something to say, and I'm going to broadcast my Glaucidia into your gills. Gills are, of course, where the respiration, where they're breathing, right? And so lots of blood, lots of great living if you're a tiny larval mollusk. Enough of the pictures and the wah. Let's, okay. Damn it. So there's a zoom in at the Glaucidia. You can see why they're named after these barbed, because they are literally Pac-Man and that little attachment thread. So kind of responding to the environment and then slamming shut. And if we play this, so there's the lure, and then boom, all of that white, that's Glaucidia, and the fish is going,
and it is infected.
And we're going to see, you can see the red of the gill underneath the cover there. Yeah. And I think the video continues till we zoom in. And you can see the little pack people, the little Glaucidia, those are the white specks there on that gill tissue that are growing in. So you've read about this, but isn't it like, it's oh my God, I'm having a hard time not swearing. It's a WTF moment.
Yeah, so cool. Okay.
So
next. So the conglutinates are these little lures, right? Little packages of Glaucidia that basically are on fishing lines made by the mantle, and then they are extended out in order to trap, to lure in the fish to then release the Glaucidia. Okay?
It's like a little glutinous Trojan horse.
Right. And so these are not little fish.
No.
These are conglutinates that have evolved to look like little fish.
So
Bananas.
the thing to remember, another thing to call attention to about the mollusks, if I can just throw it in because it blows my mind, they don't have eyes.
They don't see.
They don't see. I can see. I have eyes. If you gave me gorgeous Play-Doh
Yeah.
to make a fish lure, it would be, I would roll it out and it'd be like, here's a worm.
Check this out. So here's a conglutinate on a rock.
Delicious.
That's not a fish. But you can see the Glaucidia going through the gills, right? There we go. Infected. And the muscle is like not even there. Like it's just around the corner. There.
And there's some
Another
questions about, is the muscle sacrificing itself or sacrificing part of its mantle? It doesn't lose it.
No.
That's kind of the amazing thing. It might get nibbled, but it regrows.
Yeah. It's amazing. Okay. And then the final one. This one's a little bit different. It's a little more violent.
Yeah, there's very little consent here.
Remember, you don't want to kill your host. If you're a parasite,
Yeah.
you do not want it. So
That's a
no
mistake.
fish will die
Yep.
in this particular. But this is a very different way. So they save energy on not producing those tissues through the mantle. And instead, they use their shell in order to implant their Glaucidia.
Better
You'll see.
inward. La, la, la.
Ah!
So remember we talked about sessile. They can move, and in some cases they can move quite quickly.
Yeah.
So you can see the images on the other side, on the left-hand side of the slide. There are actually, in some species, barbs so that the fish can't back out until...
Until it gets released.
Hey Fred. Hey Fred.
What's going on? Nothing
You
to
okay?
see here.
Fred?
Yeah. Hello.
Marty? Is that you?
Okay. So the fish is fine. It's a little stressed, right?
Certainly stressed.
But the fish is fine, and it will leave. There we go, with Glaucidia in its gills. Okay. Wonderful. Now it's relaxing.
I just love this.
Okay. Good.
So there's so many great questions in here. The fish get trapped. Venus flytrap. Does the
Yep.
does the fish hurt? It's an underwater Venus flytrap. Top 10 greatest pranks of all time. What are the conglutinates made of? It's basically a complex mucopolysaccharide. But look at that.
Look at that beauty. So here's the shell, right? It's a big muscle, or at least a zoom in,
Yep.
right? This is the shell. This is the
It'll be as
mantle
big as your
tissue.
palm kind of thing.
Here's the mantle tissue
Of a different one.
of a different one that has evolved to look like food, so that it can attract the host. Just mind blowing. So beautiful. Here's a top view. There we go. Isn't that cool? All of this is mantle tissue. There. Beating to look like a swimming fish.
So again, we talk about a largely sessile, not like an immobile organism, but this is one that is moving to either attract or to hold
Yeah.
its prey.
I think
it has an
somebody-
eye spot.
They can't seem to access the chat. The
Chat's
chat
on
is on
CourseLink.
CourseLink.
That's right.
Go to CourseLink, your course, Bio 1070, and then on the nav bar there's a chat, and then select Class 2.
Does the Glaucidia have any impacts on the fish's health? If it was an enormous infestation, perhaps. But again, like Dr. Jacobs said, if you're a parasite, you don't want to really adversely affect. You want to manipulate the behavior, but you don't want them to be certainly not killed.
Yes. Don't kill your host. That would be a big mistake. Right?
Wah, wah.
Okay, just in wrapping up, we see that it is 20 after 10. We have some sort of help, some guided study stuff that you can do here. Please feel free to discuss these on the discussion board. If you're looking for, sort of input from other students, I think there's a Discord for this course as well. Please, go ahead and discuss.
And maybe a snap group, but whatever that is.
I don't know what that is. But anyway, okay. What are some of the advantages and disadvantages of complexity? Okay, so these are complex lures. There are some species that have simple lures that maybe just look like algae kind of blowing in the current, right? What are some of the advantages of complexity, but what are some of the disadvantages of complexity? Think about this from an evolutionary scale, and then also think about it from like an individual scale, from a physiological or ecological. And again, like, you don't have to be able to walk away today with the answer. It's a big answer. There's a lot to talk about. We will guide you through all of that over the coming weeks. But just to give you a bit of an idea, that's where we're going. And then, more specifically, we told you about the eunyonids and the Glaucidia larvae. The zebra mussels have a completely different life cycle. They do not need a host species. They do everything on their own, and they have something called a veliger larvae. So it's a very different strategy that can really kind of explain what's going on right now in the freshwater lakes with respect to the invasive species and this kind of competition with the endemic species. So we would love for you to explore that life cycle a little bit more and to, again, compare it to the eunyonid life cycle. What are some of the benefits and some of the disadvantages or the costs associated with these different strategies? Remembering that both of these have evolved. They do exist. And therefore, there must be some benefits to each of them, right? And with that, we say thank you.
Bye-bye.
Bye-bye. If you'd like to stick around for discussions, questions after class, we're just going to take a quick break. We'll stay on Zoom, and we'll be opening it up in just a minute.
Hello, Dougie. Hello, Dougie. Dougie can accept questions. He does not have a lot of answers, but...
Okay. Closed captioning is still on, but...
Oh my goodness, it's hot in here.
Mm-hmm. But Thomas is working downstairs.
Is he?
Yeah, I think so.
I'm amazed that... That's too bad about the recording.
Sorry about that.
Yeah.
But you found it kind of at the perfect moment, because we just took up the homework, and then we just started.
Yep.
So...
You want to stop it and start that process?